nervous system is the network of nerve cells and fibers that transmit nerve impulses between the different parts of the body. It plays an important role in your everyday life. The nerve system is divided into two parts: the central nervous system, which is your brain and spinal cord, and the peripheral nervous system, which are your nerves and ganglia, which are outside the CNS. The peripheral nervous system is further split into a sensory and motor division. This motor division is split into the somatic, voluntary nervous system, and the autonomic, involuntary nervous system, which can be divided again into sympathetic and the parasympathetic nervous system.
Neurons receive and transmit information. Here is a presynaptic cell and a postsynaptic cell. The neuron's organelles are located in the cell body. Signals are received via branch extensions called dendrites. Signals are transmitted to other neurons via an axon. Signals are generated at the base of an axon at the axon hillock. The info is transmitted through a branch junction called a synapse. In the synaptic terminal, neurotransmitters pass the information from the transmitting neuron to the receiving neuron. The presynaptic cell transmits info, while the postsynaptic cell receives it. The arrows show the overall pathway of the message.
The red squares are potassium ions and the purple dots are sodium ions. Note the ion concentration gradient across the membrane. This causes a voltage gradient across the cell membranes of each cell. The difference in electrical charge is called its membrane potential. The membrane potential of a presently inactive neuron, its resting potential, is around -60 to -80 millivolts. The extracellular fluid has a much higher sodium concentration and a much lower potassium concentration. The gradient is maintained by sodium-potassium pumps in the plasma membrane. This selective permeability only allows certain ions to pass through. A resting neuron has many open potassium channels, but significantly fewer open sodium channels. Potassium ions leave the cell, leading to a hyperpolarization of the cell, an increase in the magnitude of membrane potential, by making the inside of the cell more negative. This negative charge is the source of the membrane potential. The action potential uses saltatory conduction to move through the axon. Stage 1. An action potential is generated as sodium flows inward across the membrane at one location. Stage 2. The depolarization of the action potential spreads to the next region of the membrane, reinitiating the action potential there. To the left of this region, the membrane is repolarizing as potassium ions flow outward. Stage 3. The depolarization-repolarization process is repeated in the next region of the membrane. These local ion currents drive the action potential along the length of the axon.
Step 1. Resting state. Sodium and potassium channels are closed, and no ions move in and out of these channels. Step 2. Depolarization. A stimulus will open some of the sodium channels, and the inflow of sodium through these channels depolarizes the membrane. At a certain membrane voltage, the threshold, this triggers an action potential. Step 3 is the rising phase. Most of the sodium channels are open while potassium channels remain closed. Inflow of sodium makes the inside of the membrane more positive. Step 4, the falling phase. Sodium channels become inactivated, blocking sodium flow. Potassium channels open and potassium exits the cell. The inside of the cell becomes negative again. Step 5, undershoot. Potassium channels close and sodium channels are unblocked, though not open, and the cell returns to its resting state. The action potential uses saltatory conduction to move through the axon, bordered by glial cells. Myelin sheets are produced by glial cells and cover the axon, increasing the speed of transmission and the action potential by decreasing the total number of times ion channels have to be opened and closed, and letting the action potential jump quickly from node to node, where it can be regenerated. The efficiency makes it far more space effective, as they can remain very thin and still be incredibly fast. In the peripheral nervous system, Schwann cells wrap around the axon to form myelin sheets. In the central nervous system, they become oligodendrocytes.
Here's the chemical synapse when an action potential depolarizes the plasma membrane of the synaptic terminal. Open voltage-gated calcium ion channels in the membrane create an influx of calcium cations. In the elevated concentration of calcium cations in the terminal causes synaptic vesicles to fuse with the presynaptic membrane and release neurotransmitters into the synaptic cleft via exocytosis. The neurotransmitter opens ligand-gated ion channels in the postsynaptic membrane. When the neurotransmitter leaves, the channels close. If the membrane potential reaches the threshold in the axon hillock, an action potential is generated and the signal continues along the axon.